and I'll be sharing it. And my name is Dora Tobye. I'm the technical manager for the Center for High Performance Computing. We are in Cape Town. Earlier this morning, Dr. Stoller did talk about that this conference, we had a little time to organize it. And I just want to take this opportunity to thank the people who will be presenting today, because with that very short notice, they managed to accept the invitation and come and share with us what they are doing. As an enabler of the research, I like to call our users, but from my side, they are our partners, collaborators, because we give them the resources and they help us to use those resources to fulfill mandates for South Africa, for the community. So it's very important that we work together so that we can actually contribute to South African population. I just want to take just 5 minutes. We only have 2 talks. Just to say, the NIC is loud. It is hosted in Cape Town at the Center for High Performance Computing. We have enough resources for research, academia, and other. We also have, we're working closely with researchers who work with government departments. So it's a very important asset for the country and for CHPC. We currently hosting over 50 projects. And since we started with the cloud in 2020 during COVID, so yeah, we're growing, and we like to invite everyone to come and apply for resources there. Okay, my job today is just to chair the meeting, and then I'm going to introduce our speakers. Our first speaker is Dr. Linda Maobi, from South African Medical Research Council. So he will be telling us about what he's doing and with the cloud, and then how is it important for him, and can we welcome Dr. Maobi.
So a very good morning, everyone. My name is Linda Maobi, and I'm from the South African Population Research Infrastructure Network, which is short, it is SAPRIN. We are hosted at the South African Medical Research Council, and we are fully funded by the Department of Science and Innovation. So also good morning to our funders. So today I'll be talking about our data science platform that we're intending to build. But before I go into that, I'm also going to describe what we've been doing at SAPRIN in terms of CHPC. So this is the outline of my presentation today. We'll first talk about SAPRIN. What is SAPRIN? We'll talk about the core protocol. Then I'm going to be highlighting why and how do we use SAPRIN data on CHPC. Then we're going to talk about the current state of our data repository and what we have envisioned as a future state of the SAPRIN HDSS data science platform. And then I'll briefly describe the next steps that we're going to be taking. So what is SAPRIN? So SAPRIN, like I said, stands for the South African Population Research Infrastructure Network. It was established in 2017 as part of the Department of Science and Innovation's South African Research Infrastructure Roadmap, which aims to enhance research capacity in the country. So SAPRIN is one of the 13 research infrastructures, and it falls under the Human and Society's Scientific theme. SAPRIN is also a network of health and demographic surveillance system nodes, or HDSS, that support improvements in health, social, and economic well-being of impoverished communities. Basically, an HDSS is a standardized field-based information system and research platform where we prospectively collect longitudinal data from whole populations, including individuals, as well as households within a defined geographic area. So if you look at this slide here, I don't know if you can see it, but we have a gated community there with a fence. So think of that as our HDSS, okay? Inside the HDSS, we've got locations or dwellings, and we've got households which are resident at a particular location. And then the last component is individuals, which are also members of the household. So our focus, when we talk about HDSS, is only around this gated community. What happens outside of the HDSS, we don't really focus on. Our focus is only in terms of what happens inside this HDSS. Okay. This is a dynamic cohort which changes from time to time. We can have people moving into the HDSS through in-migration. We can have people leaving the HDSS area through out-migration and even deaths. Okay. So this is the geographic coverage of Saprin HDSS. So it's not a single HDSS. We are a network of currently 6 HDSS, 3 rural nodes, as well as 3 urban nodes. Now, our oldest node is the Agincourt node, which is located northwest of where we're currently at. So it's about a 1-hour drive, and that node is called Agincourt. So they have started collecting data since 1993. And then the second oldest node is the Dimamo node, which is located near the University of Limpopo, which started in 1995 with a current population of about 80,000 individuals under surveillance. And we've got about 15,000 households, as well as just below 16,000 locations that we are under surveillance in Dimamo. The last of our rural nodes is the African Health Research Institute node, which is located in northern parts of KwaZulu-Natal. They started doing data collection in the year 2000, and it's got a current population of just under 150,000 individuals. Then we've got our newer urban nodes, the first one being GRT Inspired, which is located in Gauteng. It consists of 3 sites. The first site is Melusi, which is an informal settlement. Then we've got Attridgeville, which is a township in Pretoria, as well as inner city of Hillbrow in Johannesburg. So they've just completed their baseline with about 46,000 individuals, 21,000 households and locations were enumerated. And then we've got C#, which is located in the Western Cape. I do not have the stats for this one yet because they've just started their baseline. They are located in Bishop Lavis Township, as well as an informal settlement called Nomzamo. And the last one is Usinga, which is located near Mangosuthu University of Technology in Umlazi. They are also going to start their baseline. Okay, so that's basically our Saprin nodes in a nutshell. And we are currently looking to establish a 7th node, but discussions are still underway. An expression of interest was sent out to various scientists. So we've just received some applications, and they're busy going through an evaluation process. So the Saprin Core Protocol is basically the collection of households as well as their member residents within the HDSS areas. And the study components is firstly the household component, where we conduct 1 in-person interview per year, as well as 2 telephonic follow-up interviews. So in all, we collect data 3 times per year from a particular household. We also have the individual component, whereby individuals older than age of 15 are eligible for a 1 in-person interview per year, where we collect dry blood spot samples, as well as conduct an HIV test. And on top of that, we also take blood pressure measurements, waist, height, everything about that individual, including collecting stuff like BMI. For all deaths that occur in the node, we also do verbal autopsies on the death so that we can find out the probable cause of death as well as their contributing factors. The last component is record linkage to services. We have not yet implemented this, but it's something that we are trying to implement in the next year, whereby we're going to link individuals to various government sectors like your hospitals, your clinic records, and so forth. So this picture basically shows the longitudinal population data that we collect. Like I said, we first start in a geographically defined area where we've got a dynamic cohort, whereas individuals enter this cohort through a birth or in migration, and they leave this cohort through death as well as out migration. So far, we have collected almost 5.6 million persons years of observations from about 340 to 530 individuals in the HDSS. Now, the longitudinal data we collect is integrated around unique individuals, households, as well as homestead identifiers into a complex longitudinal relational database. Sometimes it is not practical to do statistical or data analysis directly on these databases, and it is necessary to produce data sets that are more amenable to processing, but still maintain their essential longitudinal characteristics that will enable causal inference to be made. So our approach is to basically take the episode data that is stored in our SAPRIN database that represents states such as being resident at a particular location over time or being a member of a household for a defined period of time. And we combine those with status observations such as marital status, employment, as well as HIV serostatus. We have learned that the best way to do this is to first disaggregate the data into individual days, okay? So we take that whole episodic data, which has got a start and end date, and then we disaggregate it into day level. In other words, integrating the data in such a way we can reconstruct the day-to-day life of an individual in that HDSS under observation and record everything we know about that individual at that level of detail. This day-level data can then be aggregated back into episodes during which we know all the time varying data remains constant. And these episodes can then be analyzed using techniques such as Cox regression. So if I look at this particular data set there, it's got 4 episodes for a particular individual. If I look at the first episode there, it should be an episode for residents. I know you can't see it very well here, but these oranges are the same, but these ones are a bit darker. So it basically means that for the first 2 episodes, which are the lighter ones, the particular individual was a non-resident member in that HDSS. However, for episode 3 and episode 4, they were resident. The yellow there shows the household membership, which is yellow, basically means they were not a member of that household, but everywhere you see blue, it means that they were a member of a household. So in essence, in episode 1, they were non-resident, but they were also a non-household member. but in episode 2, they were non-resident as well as a non-member of the household. So that's how you can basically interpret these episodes. So that's the basic structure of the data sets that we have in the Saprin repository. So why do we need CHPC? So obviously this process is very data and process intensive, and we do require the capabilities of the CHPC to basically transform our data into the episodic form that you've just seen. Currently, right now, we've got about 5.6 million person years of observation, which translates to 2.05 billion person days. Okay? So imagine we have to break down this data into 2.05 billion person days. And in our episodes data set, we produce about 1.18 million basic episodes. That's the number of observations which are in our episodic data set. However, if we break down our episodes further based on age and year, so basically means that when the age of an individual changes, we create a new episode, or when the start of the new year starts, then we create a new episode. That translates to about 12 million episodes that we produce. So the setup that we use on the Nikki's CHPC cloud currently is a single virtual machine which runs Windows Server 2019 with about 25 processors, about 256 gigabytes of RAM, and we've got about 1.2 terabyte of disk space, which is running out as we speak. So we use SQL Server for the input databases. We use the Julia programming language to basically do the extractions, the data quality assurance, as well as the production of our data sets. Before then, we used to use an ETL tool called Pentao, but we found out it was very, very slow in terms of producing these data sets. And lastly, we disseminate our data using the NADA repository, okay, which is a repository which was developed by the World Bank. Okay. These data sets that we disseminate through the NADA repository are then documented using DDI compliant metadata, and we do assign digital object identifiers. So the nodes, or the Saprin nodes, basically supply us with the databases, which are anonymized. So this will be the individual level type of information, the demographic information for an individual, including their status observations. However, they give us the data anonymized, and the anonymized data then goes into a standard Saprin database. So all 6 nodes will give us data in a standard form so that it's easier for us to process. From a CHP side of things, this is where things start. Remember, I talked about the SQL server. So all the data that comes from the nodes is stored in a SQL server on the CHPC cloud. We then do quality assurance. We have written some really fancy scripts there that do parallel processing to do data quality. And then we do our data set production on CHPC, and finally disseminate our data through our Saprin NADA repository, which sits on the SAMRC infrastructure. Now, this is a typical flow of our data set production. So the blue swirly thing there shows Pentao, which was our original tool that we used to create these data sets. And these 3 dots here represent the Julia programming language. Okay? Something about Julia is that it combines the syntax of Python as well as the speed of C++ and Fortran. So we found that to be a very, very efficient programming language to do these kinds of things. Now, just to give you an idea, it used to take us 6 days in total to create these data sets that I'm talking about. And by moving from Pentao to the Julia programming language, we're able to produce our yearly data set in just 2 days, and that equates for the Julia programming language being 3 times faster than creating these ETL jobs using Pentao. So we've seen this picture before. This is our current state where we have, or where we disseminate our data through the NADA repository. This is basically why I'm here today, and this is part of what we're trying to achieve at Saprin in terms of removing this NADA repository, because as it stands, the repository works, yes. You can go and log into the repository. You can download our data, which is publicly available. However, we found that for most of our users, especially from the public health side of things, they don't know how to analyze this data, or they're having difficulties in terms of analysis. So what we've envisioned is a whole new different type of platform, which not only gives you access to the data, but also gives you access to a whole community whereby people can contribute in terms of scripts or in terms of analysis methods for Saprin data. We are also envisioning a new platform whereby we can also post tutorials, video tutorials, for our users in case they want to learn more on terms of how to analyze Saprin data. So our future state looks like this. We're now going from just a data repository whereby you can download data to a more of a community-centric data science platform. And we've explored a couple of possible solutions in terms of doing this. The first of these is CKAN, which stands for Comprehensive Knowledge Archive Network, which is the world's leading open source data science platform. The nicest thing about CKAN is that it is extensible. You can do much more than what you can do with NADA. NADA has got a lot of limitation when it comes to customizations and so forth. But with CKAN, you are able to create custom plugins and increase what you call this. You've got a full API integration as well as visualization tools and more. The other solution that we're looking at as well is Jupyter Hub, where we want to give users access to computational environments and resources without burdening the users with installation and maintenance tasks. So this is where we are going to basically integrate Jupyter Hub with our Saprin database and also put in predefined analysis that you can already run on Jupyter Hub to get your results right then and there. So we want to cater for our public health specialists who want to understand mortality trends in our nodes, but not necessarily know how to write code. So these are some of the high-level requirements we've discussed with the CHPC. So we've been in constant communication with Dr. Anwar Vahed, with Dora, as well as Ingwe, in terms of our functional requirements for this new data science platform. So these are not all the requirements yet. It's not a comprehensive list, but I've just highlighted one of the most important ones. So the first one I want to talk about is data storage, which is functional requirement number 2, which is the ability to upload, update, and delete microdata. So this is the current functionality that NADA is giving us at the moment. However, we still want that functionality to cross over to the new data science platform. The other one, which is very important, is the Saprin data analysis. We want our users to be able to log into that data science platform. They already have the scripts written out for them. All they just do is run whatever analysis they want to run, and then they get the results right there and there. The other one is Saprin video training guides. So as Saprin, we've been going around different universities training students as well as researchers in terms of analyzing longitudinal data. The most recent of these, we were at the School of Public Health at the University of Pretoria, where we delivered a course on event history analysis or survival analysis. We want to incorporate all these trainings into our platform so that anyone who is interested in doing this type of analysis can have access right then and there. The other thing that we want is functional requirement number 8, which is collaboration and sharing. We want to form a community around the Saprin data platform, whereby if somebody's looking for, let's say, a calculation for fertility rates, we might have missed it out in terms of putting the script there, then somebody else can contribute to building that script for us that will run on the Jupyter notebooks. So these are the next steps that we want to take. We've got a couple of phases. I've just highlighted only the first 4 phases, which is just the initiation, planning, design, execution, as well as testing and enablement. So in the initiation and planning, the first thing that we needed to do was to get a development environment, of which the CHPC was so kind enough to provision for us. So that one is complete at this moment, and this environment will then enable us to move over with the next steps. Now, we're also looking to mobilize resources for this because we'll need people to develop this system. So this will be a collaboration between CHPC, DERISA, as well as Saprin. And then early in the next year, we are looking at going through the business requirements workshops so that everybody is aligned to the goals that we want to get out of this data science platform. Then we'll start with the design, maybe in Q1 of next year, and then start with the execution, which will involve installation and configuration of CCAN, as well as the Jupyter Hub that we require. And then we're going to be moving forward with the integration between Jupyter Hub and the Saprin database, configuration of CCAN as a data repository, and then theming development for CCAN. After all of this is done, I'm hoping that by the beginning of Q2, quarter 2 of 2024, we'll get to the stage where we do user acceptance testing and then create user training documents. Of course, there's other steps which are involved there, such as deployment to production and user acceptance testing later, further down in production. But for now, I'm just highlighting what we can do right now in Q1 of 2024, going into Q2 of 2024. So this brings the end to my talk, and I'd like to thank DERISA as well as our CHPC colleagues for being supportive of this Saprin data science platform that we want to take forward. A special word of thanks to Dr. Anwar Vahed, who's been extremely supportive in this. I'd like to thank Dora for all the assistance as well as technical support in this, as well as Ingwe. Thank you, sir, for much of your assistance during these times and for being there whenever we need support. Thank you, everyone.
Very sensitive data here on households and their health and all of those things. So I presume that before having access to this data, even in its processed form, there might need to be some kind of control of data access. Have you considered flipping the model where you create a trusted research environment where what people submit is computational queries rather than giving them access to all of the data that in the system, and then they obviously have to have a data access agreement. So in other words, building a trusted research environment, because that is the field, that is the direction which I see a lot of the field internationally is moving in.
Thank you. Your name is?
Sorry. I'm Peter from South African National Bioinformatics Institute.
All right. Thank you, Peter. Thank you, Peter. So that's a very good question. So the data that we currently have in terms of our database is fully anonymized. So we don't get any sensitive data from the nodes. we make sure that all identifiers that point to a particular individual are removed. We also do statistical disclosure control to make sure that quasi-identifiers are also removed. And we've got various thresholds that we use. So what we receive from our nodal partners is a fully de-identified data set. And from there, we don't have the problem of sharing the data. So yeah, we don't have any issues around sharing data. There are data agreements. There's various methods that we've put in place to make sure that the data from individuals is not exposed. Thank you.
I think there's a question there. Rama?
Yes, thanks for the opportunity. I just wanted to ask you, you just said the data is anonymized to the level of removing the quasi-identifiers. So doesn't that affect the level of what you can do with the data? And if it does, to what extent? Because the more you anonymize, the more it becomes generalized data. So for example, in one of the clusters you showed that has households, then if you want to pinpoint maybe a household, does that mean you now give them a different data set, or depending on how you want to use it? Because now the health practitioners maybe want to pinpoint somewhere, but now it's just generalized data. How do you deal with that?
Right, so that one we don't deal with it at the SAPRIN level. It's dealt at the nodal level. But what I can assure you is that the number of individuals who have got like quasi-identifiers that might lead, it's very, very small. It's like 0.000000% as compared to the entire population. So I wouldn't say that will sort of like influence your research or in whatever way. It's very, very small amounts that we've seen. Yeah.
Any other questions? Okay, there's a question there, sir.
Okay.
Hi, thank you for your presentation. I'm Ginelwe. I'm from the South African Environmental Observation Network, and we also do host various research infrastructures. My question to you is, how interoperable is your systems architecture and metadata with other infrastructures within the South African infrastructure roadmap? Basically, what I'm trying to ask is that I see some of the data that you have collected being useful for our data portal as well, but I want to know, like, in terms of your metadata and systems architecture, are they interoperable so that maybe we could chat afterwards and try to...
Are you from Seyon?
Yes.
Okay, cool. That's a very good question, because we have actually started a collaboration with FTON, led by Dr. Gregor Fake. At this moment, we're still trying to explore avenues for collaboration. I can tell you that in the last month, they have implemented one of their, environmental surveillance node at one of our SAPRIN sites, which is Agincourt. So at this current moment, we are basically, at the stage whereby we want to figure out how we can work together. So we have not yet finalized the details, but there is an ongoing working group meeting for SAPRIN climate health type of data, whereby FTON is supporting on the climate side of things, and we coming with the data aspect of it. So definitely there is collaboration between FTON and SAPRIN at this current moment. Sure. All right.
Hi there, this is Uljana Hesse from the University of the Western Cape. How is the data used? I'm not, what analysis, what are the interpretations what do you get out of the data, and how is this used to promote the life of the people that actually gave you all the information? That's one question. And the other one is, what is the age limit of a person that participates in this study?
Okay. All right, so in terms of age limits, I'll start with that one. It's easy. So everyone is included into the study. However, for the individual component, whereby an individual has to basically respond for themselves in terms of the questions asked, it's only individuals who are 15 and higher. All others who are underage, their mother or their guardian is the one who's responsible for giving out the information. So for instance, we'll record someone from the day they were born up until the day they die, okay? Including their birth weight at hospital. We'll receive all that information from the Road to Health card, the Road to Health card. So we'll receive all that information and it is being recorded, okay? So everyone who is part of the HDSS area, who lives in the HDSS area or is a household member, whether they are resident or non-resident. So what do I mean by resident? A resident household member basically means it's somebody who lives in the HDSS area. But then you find out that certain individuals are still members of the household, but they work somewhere else in Johannesburg. They are still eligible to be under surveillance as long as they still retain their household membership. So everyone in that HDSS area is eligible for surveillance. And we've got about above 95% participation in all the nodes. Okay, so it's just a few who might refuse to be under surveillance, but those are very rare cases. However, we started seeing them in the urban nodes, because the urban population is quite different from the rural population. Then your other question was around what do you use for this data? So most of it is being used from a research perspective in terms of understanding epidemiology, disease surveillance, and so forth. For instance, during the COVID-19 pandemic, we did do tests on individuals living in that area just to understand disease progression as well as access to health facilities and so forth. So all of this information, we use it for different types of studies. For instance, in some of the nodes, apart from the demographic surveillance, they also run clinical trials. So because we've got this rich cohort of individuals living all in that geographic area, if, let's say, they want to run a clinical trial for specific individuals with certain characteristics, you can go through that database and extract all those individuals. And then, obviously, you need to get consent from them before you can run any trial. So the research platform is not just about demographic surveillance. There's also other things that happen, like, for instance, embedded studies. If you want to understand health utilization for sexual and reproductive health, those are some kind of research that happens in the nodes.
So that, I'm sorry, but then I have to continue. For example, when you are doing health-based analysis, then you depend on, in order to identify places where you have the, for example, an upflare of COVID, you depend on the localization, which means you need to know the individuals which are associated with a specific localization. So how can you still speak about anonymity of the data if you say, for example, in this town or in this community or in this house, you are going to see COVID cases? So if you are talking about the analysis of a network of a, help me out with the language. So when you are looking at an aerial distribution, you will have to see, if you look at households, you will have to look into where they are located. And based on that, then you can backtrack the people. And then your anonymization is breached. Does this make sense?
No, I mean, the thing is, remember, we are covering an area of, let me give an example, 850 square kilometers. for one of the nodes, right? So when we go to households, we have GPS locations for each and every household. And those are not shared, okay? So we do have internal identifiers for each and every individual. That could be their South African ID or their passport if they're not South African. So we do have internal identifiers to accurately capture specific individuals within the HDSS area. What we share is the anonymized version. So for instance, you can have an internal ID called 881230. But what we'll share to the public for that specific individual will be ID1 or ID2 or ID3. So we remove the names of individuals, their surnames, and you can only identify them using a unique external identifier like 1, 2, 3, 4, 5. So what we have internally is different from what we give out to the general public. So even in the case of locations, we do have the GPS coordinates of each and every location that we visit. However, when we share the data set with you, we'll say this individual one lived at location 10 and is part of household number 12. So there's no way you can actually go back and say, what is household number 12? Yeah. So we've fully de-identified the data. Any other questions?
Sorry, just following up on the geospatial thing, and sorry, I'm on the other side of the camera, but yeah. When I was working in the Western Cape on trying to assist the contact tracing when it was still being done, then one of the things that I noticed was that if you use the standard geospatial resources, they don't describe places like, for instance, in Cape Town we have an area called West Bank. It simply does not exist in the official GIS resources. There's a West Bank in Malmesbury, which causes great confusion. So do you also feed back, when you are finding out, for instance, that people describe their area in different ways? I mean, because can you be part of, or not you, but your nodes, can they be part of, actually capturing how people are describing the geography of their own areas? Because it tends to be better for the formal areas and worse for, in Khayelitsha, SST section is listed as Silvertown. Nobody there calls it Silvertown. But it doesn't seem that there's any, the experts sitting at a distance don't seem to have a connection with the voices of the people who are living in those areas. So I don't know, I mean, this is obviously more of a node level question, but do you guys get involved in capturing people's own understanding of their geography? Because that's quite important in understanding the epidemiology of things when, if you're going to start allocating people from West Bank and Cape Town to West Bank and Malmesbury, it's going to cause somewhat chaos.
So yes, that's a very good question, and you're quite right. That happens at the nodal level, of which I'm not involved in the day-to-day stuff. But I can tell you that we have community engagements in all of our nodes. So those will be responsible for engaging with the community from time to time, especially getting buy-in for various studies that we want to run there. And especially, it's crucial when we want to establish a new node like Usinga and C#, whereby the community engagement officers will proactively engage with community leaders in those spaces. However, the nitty-gritty details, I don't have them, fortunately.
I think we taking our second speaker. So if maybe you have more questions for Dr. Baoyi, you can maybe meet him outside and discuss further.